Good evening, guests. Welcome to Prof. Roets' inaugural lecture. Can you please stand for the academic procession as they will enter? Please be seated.
Good evening, everybody. My name is Dani Brink. I'm Dean of the Faculty of Agri-Sciences, and on behalf of the University and the faculty, the department, I would like to welcome you at the event, the inaugural lecture of Professor Francois Roets, particularly his close family and friends and colleagues, both at the university and beyond, and also all the members of the public. And I can also see some of the department students also in the audience. This is actually the VIP guests, what it is all about, so thank you for availing time. I would also like to recognize Professor Stan Duplessis, the Chief Operating Officer of the University, representing the Rectorate, and also Professor Karen Sisler, who joined us in the procession as the previous head of the department, that gladly, if not graciously, handed over the reins to Francois not so long ago. I would like to just pick out a few interesting things from Francois' bio that has been also published as part of the proceedings. After completing his PhD studies at the Department of Botany and Zoology here at Stellenbosch in 2006, Francois went the typical classical route of taking up a postdoctorate at the University of Pretoria in the Agriculture Biotechnology Institute, the so-called FABI. Not long after that, he joined Stellenbosch University in a lecturing position at the Department of Conservation, Ecology, Entomology, and subsequently Francois has excelled not only in academia but also in the science domain, and he has also accepted the responsibility of becoming head of the department in 2001, so it's quite a steep curve and a very successful one to say the least. And also he remains a core member of the Department of Science and Innovation NRF Center of Excellence in Tree Health Biotechnology, which he has been a collaborator since 2008. Apart from multiple participations in international conferences, national networks, in the process he graduated 28 master students, 15 PhD students to date. He has a long record of peer-reviewed scientific publications of over 140, and he's also been recognized as Stellenbosch for research excellence in a number of years, 4 in total, and as early as 2019 and as late as 2021, where the university have recognized him for the high level of research output that he has delivered. So we really look forward to Francois presenting a flyover of his career, highlighting matters of particular importance to him and I believe also to the audience. So Francois, I would like to invite you and to come forward and proceed with delivering your inaugural lecture. Thank you very much.
Right. Thank you, Donnie, and Stan, Karen, and everybody that's here. It's a massive crowd. Didn't expect that. Everybody online, hi guys. Yeah, so thank you very much for the opportunity to present what I've been doing up till now. It's been quite a journey, but it's been a fun journey, and I hope it's not the last you'll see of me. So the topic of my talk is life took over the globe through combat and networking, and the reason for that is that I focused quite a lot on interactions between different organisms, and a lot of these interactions are good, and the organisms like each other, and a lot of these interactions are not. And life moved from the water onto land probably because of competition and those sort of things in any case, trying to get away from competitors. So evolution on the planet has been chased or pushed by these interactions. So right at the beginning when I started my journey in my first year, actually before that, I had a bit of a conundrum. I didn't know what I wanted to study. I like insects or arthropods, I love plants, and then later on learned all things about microbes, and I thought, ooh, that's also interesting. But I didn't know which one to focus on. And then sort of throughout my third year and fourth year, yes, that wasn't a planned fourth year. In the fourth year, I sort of realized, maybe I can combine all of these into one aspect. And luckily, projects came my road that helped me to do that. So the basis of what I'm interested in is that no organism lives in isolation. Every single living thing on this planet interacts with another living thing, or everything that I can think of. So on the left-hand side there, that sort of grey blob that you see there, I don't know if anybody of you know what that is. You all should know it. They're buddies of yours. These are the follicle mites that live on your face. So we all have them. There's about 60 species of them all around the world. There's probably multiple species on your face right now. Please don't poison yourself. And so there's one of these interactions that we have with other organisms. That's a different kingdom of life. It's actually closely related to spiders and that sort of things. They've got 8 legs. And they come out at night out of your pores, feed on oil and dead skin and mate skin and do all of those sort of things, and then they crawl back. So you're not living in isolation. There's bugs on you. Something like the letters on your plate. If you go to a restaurant or buy it at Woolies, no matter where you're buying it, you're not just eating one species, no matter what you do to that lettuce. That thing has a lot of microbes living inside of that leaf. Probably 100 species or more of different bacteria, different fungi living inside that plant. Most of them, we don't even know what they do. Also, most of them, we don't know what they are even. We haven't seen them because a lot of them we can't even grow. They need the plant to live in. And some of them actually help the plant. They make compounds and things that help the plant to become more drought resistant or herbivore compounds and things like that. So when you eat lettuce, you're eating a couple of different kingdoms. You're not eating only that. And then on the far right, back to our human bodies. Again, we live with microbes. So it's not just the kingdom of the arthropods, but also all of these other kingdoms of microbes that live inside us. There are actually more microbial cells in and on your body than human cells. And if you get rid of them all, you will probably die. So the problem with that is then what is a human? Is it just the DNA that makes us human, or is it that entire collection of all these microbes and our human cells that make us human? In fact, more than 1 kilogram of your body weight are microbes, roughly about 1 kilogram. So next time you climb on the scale, just subtract that kilo and you'll feel a little bit better about yourself. So those that know me and know what I do, know that I do quite a wide variety of things. And I thought the best way to run through sort of a picture view of what I do is just to have 3 examples of systems that I've worked on right from the beginning. 2 of them from the beginning and 1 later on. And the very first one is where I started my master's and PhD work on, working on proteas. So for those that don't know, that is our national flower, the protea. Beautiful plant, but it hides many, many secrets within. So on the left-hand side, there's a suikerkan, one of the Proteus species. You'll see the nice white flowers open for pollination by insects and birds. After pollination, those flowers close up and form these cones that you see on the bushes. So there's one sort of in the middle of that plant over there. And the idea of that is to keep the seeds safe until there's fire around. So when the fire comes through the system, those cones open up and the seeds are dispersed into an area where there's no competition with other plants. Free of competition, lots of food, lots of nutrients hanging around. But during that time, when these cones are on those plants, they actually get occupied by a lot of organisms, a lot of insects and other things. But for me, most intriguingly is a fungus, or a whole group of fungus called the ophistomatoid fungi. So you see these little black knobs, things that you see there, that's on the flower sort of parts inside of these cones, and they dominate this whole system. So if they are there, they are the most common thing in terms of microbes there. And what's interesting about these guys, well, one of the interesting things is that usually their sort of relatives, close relatives, associate with beetles or things in the northern hemisphere that cause plant death and destruction, so diseases and things like that. Here in Proteus, we find them in flowers. They don't seem to cause any disease, and the beetles aren't there. So what is going on in Proteus? How do they get around? So on the right-hand picture there, you can see that's a tip of one of these structures where you can see the sticky spores. So the spores collect in the sticky drop. So if you look at those structures, the spores are produced inside that base at the bottom, and then pushed through this long neck, and then collect at the tip there. And right at the tip is in a position where anything that walks past, or insect or something that walks past, can get into contact with these spores and pick them up and transport them to a new host. So the obvious thing here was to look for some other insect or arthropod that would transport this. And this is important for the fungus. You're inside a closed cone. There's no wind, there's no water, there's nothing else that can disperse you. So you need the help to get out of there. So the first sort of eureka moment, after lots of failed attempts of finding who the culprits are, we've actually found a couple of mite species that carry these fungal spores. So the mite in number A there, the arrows there point to pockets associated with the legs of these mites in which these spores are carried. So they feed on the fungus and they carry the fungus. At the bottom side there, the mite there, the poor thing has been squished on a microscope slide, but you can see the fungal spores sort of on the sides of its body. This mite has these folds inside of on its body, and it actually packs the spores inside these folds. And why do they do that? Well, we could do some tests, put some mites on petri dishes where we grow the fungus, put the mites on there and see if they actually do anything with the fungus. And lo and behold, if you look at, I don't know if there's a pointer on this thing. Yeah, okay, it's not going to help much. So the darker bars that you see there are the fungal spores that they carry. And if you put them on those same fungi, they actually increase their population sizes quite drastically, significantly. So they feed and reproduce by eating the fungi that they carry. So they carry this fungi between different plants. They take the fungus to a new inflorescence or infrutescence, a flower structure, and they actively farm this fungus. So they're the farmers of the mite world. They feed on these fungi, which is, everything is great. I love that idea, except after the massive fires we had in the famous sort of brought me to a realization, well, what happens after a fire? I mean, these mites can't fly. So suddenly you have this, basically a desert, all the proteas are gone. It's going to take at least 5-6 years before the first flowers appear again. So how do the mites get back there? Mites can't fly. Well, actually they can. They just climb onto pollinators of these proteas. So on these pictures, those little white dots that you see, it's on the other side of that beetle, of these mites that's clinging to a beetle, that feeds on the pollen and nectar of these plants. And also in that sort of V at the top of the beetle, you can see some of the mites stuck in there. So these mites are quite clever. They can't fly, but they climb onto these taxis that take them around. And then, I think it was Janneke, with her study, threw over the apple cart a little bit. She did some population genetics on the fungi. I don't expect everyone to understand what's going on here. Basically, each dot that you see there is a different individual or group of individual, and the different colors are where they were collected from, and the little lines connecting them tells you something about how related these are genetically. What you'd expect from a population study usually is that all the colors would group together because they come from the same area. All the samples from one location would be kind of the same. What we see in these networks is that there's nothing, no pattern whatsoever. And if you look at the key at the bottom there's populations from Betty's Bay, Tuticoma, Neotville. This is across the whole Western Cape. So how do the fungi travel from Betty's Bay to Neotville to Tuticoma in such a way that you can actually map it quite effectively like this? The insects can't do that. The insects definitely do not fly that far. There are mountains in the way, all kinds of other things. So it was back to the drawing board to see what is actually going on here. So there we have our proteus. So I had to take a much closer look than before at the protea. That's a protea nereifolia. If you have not dealt with proteas before, never sniff a protea, especially in the field. If you touch them, you'll see all of these mites running out, called protea itch mites, and that's for a reason. They do make you itchy. So those mites are quite large mites. You can see them with the naked eye. They feed on nectar and pollen, and you find them in almost all the proteas. But what our fungus-carrying mite does is very clever. It climbs on top of these mites. So you can see in that picture there's a small little bump on the bigger mite. That's a small mite that carries the fungus that climbs onto this bigger mite that is in search of flowers for pollination or for pollen and nectar. And that big mite is the clever one. That guy uses birds to travel around. So what we have in the system is a fungus-eating mite that climbs onto a pollen and nectar-feeding mite, and that mite climbs onto birds for this massive long-distance dispersal, which explains how the genetic material can travel throughout the whole Western Cape. This is a graph showing that how many proportion of the mites that actually carry the fungus, and some of these mites, almost all of them, carry this fungus. So why is the population? So that's one of the reasons why the population diversity is so high. These things just move around quite easily. Lots of fungal spores, lots of mites. Things are going everywhere, almost simultaneously. But another thing, again, Janneke, what she was looking at, she looked at the genomes of these fungi and looking at the mating type genes. I don't expect anybody to understand this. I barely do. But looking at the mating type genes, we could see that the fungi is actually heterophilic, which means you need two different strains to come together with opposite genotypes for this mating type strain, for this mating type gene, and they can combine or recombine and basically have sexual reproduction, which then enhances the ability to produce more genetic diversity, continuous genetic diversity. And you remember all those structures that we saw, those dots in those flowers, each one of those is a sexual structure. So that's the sort of result of this recombination happening. So when I started off these studies, we had 4 different species in just one genus. We're up to 12 different species, only from Protea. So it shows you how little we actually know of what is going on out there. And just by looking at the genetics of the fungi in the Proteus, we see some interesting things. One of the interesting things for me is that we know the genus Protea started off, evolved in the Cape, and then moved to the summer rainfall areas and up into the tropics. If you look at the fungi, they did exactly the opposite. They jumped onto Protea somewhere in the summer rainfall region and then moved down with the birds and the insects and things probably into the Cape. So what do they do in the Proteus? I mean, they're dominant there. This was also, I think it's a whole PhD almost worth of work, if not more. So from the genomes, but also from growing these fungi on different media types, we can understand what the substrates are that they use. And at the very early stages of the fungal growth, they probably use the nectar sugars in these Proteas. So remember, they use the pollinators to travel around. So when they get to the flowers, the most energy-rich substance for them in those flowers are the proteonectars, and they actually use that and grow very rapidly. Then later on, when the flowers close, there's no more nectar, they switch modes, and they actually start degrading the cell wall compounds of these plants. Quite a nifty system. But to find out what they do there, so we did some competition studies with of these protea-associated stomatoid fungi, these mite fungi, and other fungi that we find in these flowers. So this particular example is a penicillium or a cladosporium. I can't remember which one that is. And these fungi actively break down plant cells. So if these fungi take over within the seed cones open up and they lose their seeds before there's a fire. So before they actually are supposed to spread the seeds. So what these fungi do is they actually get in there quite early, take over the whole niche, and then prevent other fungi from growing inside these flower heads. So it's actually also a mutualism, not just between the fungus and the mite, but also between the fungus and the plant in this case. So that was proteus, but all the different tools we learned there we can also apply in agriculture. This is one of the examples there. So this is a study that I did, or I was invited to take part in with colleagues from plant pathology department here and colleagues at the ARC. And it's looking at the dispersal of grapevine trunk disease pathogens. So it was assumed that these pathogens get around by wind or water. But we also saw that they rapidly expand their ranges in these vineyards. And after a lot of testing and you can imagine searching for what is actually happening here, we found out that a lot of insects can transport these fungal spores in these vineyards, mostly at night when we don't see what's happening. So that's why it was missed probably. So when these grapevines are pruned, they form a little droplet at the top of a nice sugary substance. And that's very attractive for things like millipedes. So there's a millipede at the top there. I mean, these things usually we find only on the ground surface. They don't really like to climb. At night, they climb up to get to the sugars. And as they climb up, they walk across these infected plants, pick up the spores. They also feed sometimes on these infected areas, and they transfer the spores directly to these pruning wounds. So exactly where the fungus can infect these things. We also found these spores transmitted by things like spiders, believe it or not, and ants. You can see a bunch of angry ants there that were bothered in the middle of the night. And the same sort of protocols of things we use to follow what's happening in the proteus system, you also use in these sort of systems. Right, so the second system that I'll briefly touch on, I wasn't sure about that title for it. Can plants get sexually transmitted diseases? So we're tracing here basically a body snatcher. I have to show off with this picture. This is a really nice oxalis species named after Professor Dreyer. It's a group of plants that she absolutely loves, and she's built her entire career on it. Anybody that she comes into contact with gets enthusiastic about this plant genus. And I've also been lucky enough to go on many a hunt to find these flowers in the middle of the winter, sometimes extremely cold. You go into the Richtersveld, very cold, very barren, and you just see these little plants popping up, if you're lucky. So I was involved in discovering and helping to describe quite a number of these species. But while on these adventures, we also noticed that some flowers look a little bit different than the usual ones. So if you look at the two little pictures on the right there, the one at the top, you can see the nice yellow anthers, the normal thing that you would see in a flower. The one at the bottom, you see, oops, there's something going on. Why is it that kind of mustard colored? Something odd is going on there. So in this picture, number A there, that's a healthy flower. And the one number B there is one that's obviously something wrong with. If you dissect those, you can see the one in picture in C, that's normal yellow pollen grains that you see on those anthers. And in D, what you see there is actually fungal spores that's taken over the anthers of these plants. Otherwise, the plant looks very similar. If you look, all of these photos at the bottom, these are pictures of the actual fungal spores. So even the spores mimic what pollen grains should look like. And obviously, well, if you're a fungus, you can use the plant's pollinators to transmit. So I think that's why I'm justified to say it's a sexually transmitted disease for the plants. So there's a beetle and a bee, and they're just covered in these fungal spores. So they visit these flowers thinking they're going to get nectar and pollen there, but they don't. So there's a picture of a bee that's been bamboozled into thinking that that's a healthy flower. That bee, if you see sort of at his face there, you can see the anthers there. You can see the fungal spores there. And it's actually collecting those fungal spores as if it is pollen grains. It's also taking that back to the nest, as it would with pollen grains. But otherwise, other than that, you don't see much wrong with these plants. Except if you do crossing experiments, you can see that the plants are actually sterile. So this fungus sterilizes the plant completely. And if you take the bulbs, we surface sterilize those. So these plants make bulbs. And wait for the next season for them to come up again. That plant is still diseased. So this fungus is a body snatcher. It grows throughout the entire plant, sterilizes the plant, and then forces this plant to replace its pollen with its own fungal spores. So the entire plant is basically just like a glove for this fungus. And these plants can live a very long time. Leon, correct me if I'm wrong, but there's plants almost 30 years old, in pots in the botanical garden here. So these plants live a very long time. Which means if this fungus goes on and on and on in populations, it'll take over populations. So there must be a balance somewhere in the system. And being an ecologist, we get interested in these sort of sidetrack things. And we wanted to see, well, what happens in these populations. So we found a really good use for beer boxes after the contents have been disposed of in a nice way. You actually can take the flowers of these plants before they get pollinated or visited by insects, put them in little Eppendorf tubes with a little bit of water, and arrange them in these boxes. And they can open and close and do their thing for quite a few days after that. So you can actually manipulate experimentally the density of flowers or diseases or all kinds of things that you do or want to test. So on that box, for instance, we have a bunch of flowers, some healthy flowers and some diseased flowers, and we can vary the sort of proportions there to see if the disease is more frequent in that population. If more people are sick in this room, are you more likely to get sick? It's basically what we're testing. Same with the flowers. And this is just the bees spreading that disease. And what we see in the graphs there for pollen, as the disease number of flowers increase there, the number of pollen that you receive reduces. So seed set and those things would be reduced. But also the number of fungal spores increases. So if there's a lot of sick people around you, the chance that you're going to get sick is good. I shouldn't say because I'm sick. But you can also look at the effect of host density. So it can be 100 people but spaced far apart, or 100 people very close to each other. Does that not also affect how easily you will get that disease? So this is working with plants, but it also actually applies to a lot of other disease symptoms, even in humans, things like COVID. So you can vary the interspacing distances of these flowers. So here we have the same sort of flowers in their little tubes, but now we put them on little pegs, and we can put them in a field where there's no competition from other flowers and see what happens to that. So it's vary that distances, the density of the flowers, keep the frequency of the disease constant, so say half of those flowers have that. Wait for 3 days and see what happens. So in this case, what we see is if the flowers are very dense, they do get pollinated, but not as great if the flower density is a little bit further spaced. So there's an optimum for the pollinators to do their job. If you look at the spores, similar sort of things. After 200 centimeters, the number of fungal spores that you'll receive drops drastically. So in populations where this fungus is, it would be really good if you're not bunched up together and not a lot of you are sick. if you want to prevent getting this disease. But we also wanted to know, is there some evolutionary impact of this fungus in populations? So if you have a population where there's a disease, do the healthy plants in that population, do they look different than healthy plants in populations where there's no disease? Is there some other factor influencing their evolution? So for that, we have a couple of assumptions here, and the one is that larger flowers are better. And that generally is the case for bees. Bees like showy flowers, bigger signal. So large flowers attract bees better than small flowers. It depends on if it's a bunch or single flowers, but in this case, single flowers. But if you attract bees easier, then you can also get the disease easier. So there must be this evolutionary trade-off, sort of an optimal size to make sure that you get pollinated, but also not get the disease. And the way to test this type of hypothesis is to go to diseased populations and healthy populations of the plants, find the healthy plants, and then compare them and see what's happened over time already. So that's what we did in this graph. So this is from a couple of populations here. The grey bar there is the plants from the diseased population, and you can see that their flowers are smaller, significantly smaller than the flowers in the healthy populations. But the leaf size of these plants didn't differ. Important, which means that whatever is making flowers larger in the one population has something to do with the flower, not with the environment. It's not the entire plant that's bigger, it's just the flowers. Back to our beer box experiments, and what you can do here is have the big flowers and the small flowers and a couple of diseased flowers on that box. Let the bees do their thing. So we're expecting, based on what we just saw, that the bigger flowers on that box are going to get more pollen grains, but they're also going to get more of the disease. Look at the pollen grains and say, yeah, bingo, exactly what we expect. The bigger flowers get more pollen. Problem comes in, oops, spores not. So what's happening here? Why did all the flowers get the same number of spores? And the only explanation I could think of at that stage is if bees have preferences. That makes sense. So if bee individuals have preferences. If a bee only goes for a healthy flower, then it'll go from the one small healthy flower and then choose a bigger one probably after that. If they only go for diseased flowers, they'll stick to the ones with the fungus in them and by mistake maybe transfer spores to any one of the other two, because it's by chance. So, and there's a nice way to test that. You take a bottle of wine, go sit in a field with a lot of flowers, and look at bees for many, many hours. It's tough work, but somebody has to do it. And you literally look at what the bees are doing in that population. It's sitting in a healthy flower, then a diseased one, and skipping a healthy one, and you follow them around for hours and hours and hours. You go back to the lab, analyze the data, and what you see there is, as a species, the bees have no preference. So if you're in a field with a lot of flowers, you'll count the same number of bees on the diseased flowers as you would on the healthy flowers. But when you start following the individuals, you'll see that the individual bees do have a preference. So if the individual bee started off by foraging on one that has the fungus on it, it's going to stick to that. Every now and then it'll make a mistake, but it sticks to that. If it's on a healthy flower, it's going to stick to that. So this is the reason probably why this fungus doesn't take over the entire population. It's mostly mistakes in these transfers that lead to these infections. So it was a long journey to try and figure out, but why? So there's a lot of ongoing studies, a lot of plans still to do. So there's about 20 species of oxalis that we know of that have these fungi, but currently we've only described one species of the fungus. And if we look at the data in the field, that makes very little sense. So this is some data that Andre collected a couple of years ago for his fourth year project. And again, beer box experiments, I love these things. In this case, you put some diseased flowers on a box and mix that with all the other flowers of species that you find in that population. 7 or 8 species of oxalis you could find in the same area, flowering at the same time. So mix all the flowers in there and wait for the bees to do their thing, and then you go count the spores and the pollen. So in terms of the spores here, you can see there's 3 different sites there, a couple of different species. So Oxalis lanata is the one that usually have the disease. None of the other species have the disease in these populations. In other populations, they can have this disease. And we can see that the spores get transferred to basically all the species. But in those populations, the other species don't get sick. So that means there's some other genetic sort of a signal between the fungus and the plant. So there's some speciation or something going on. We've looked at a whole lot of genetic markers, and we can't find anything variable enough to show us that there's an actual difference there. So it's still up in the air at this stage. Are these things different species? Or is there something else going on? And we'll have to probably sequence the genome of 1 or 2 of them before we can actually start answering these questions. But that's not the only fungi things. There goes my voice. That we're doing at the moment. Throughout my career and ongoing into the future, there will be lots and lots of work on pathogenic fungi. We're finding a lot of new things in the fynbos in South Africa, undescribed fungi, and a lot of them are causing problems. Currently, Janneke is working with a whole bunch of students, a whole group of people on cypress canker, which in the picture there we found on native Woodringtonia trees. So this is an invasive fungi or exotic fungi that for the first time seems to have jumped onto native fynbos plants. And of course, this particular Woodringtonia notiflora is quite widespread, but then we also have the Klein William cedar trees, which are not that widespread and endangered. So what happens if this thing gets to those populations? Working in plantations on things like eucalyptus and Terratus phorea. Lots of genome work there, lots of pathogenicity work being conducted there. Sticking to native things, newly described fungi, Immersioporthae and Diaporthae, these 2 fungi are on native plants. We don't know if these fungi are native or exotic, but they're killing a large number of trees. So looking at population genetics of these things and trying to figure out where are they from and what they're going to do. And lots of other ones. So this is a really messy slide, sorry. I don't know what I was thinking. So sort of, as I mentioned, I'm doing also a whole bunch of other things. So I'm looking at some symbiotic systems here, but I also do a lot of work on altered landscapes, a little bit of landscape ecology, a little bit of invasive things. A lot of the fungi that we work with are invasive. So a lot of things that we're looking at or have looked at is basically how our native insects and fungi can use exotic plants to move around in the landscape, which they couldn't do before. So these invasive plants are connecting populations of things that should not have been connected before. We can restore some of these habitats after we remove invasive plants, for instance. But there's also often legacy effects. So once you do restoration, you make everything ideal for pests and pathogens and things to come in and just cause havoc in these systems. We make it easy for them. We focus a lot on urban areas because a lot of the invasive things, the first time we see them is in urban environments, like Phytophthora, which literally means plant destroyer if you translate that into English. Working in interesting places, radioactive waste disposal sites, looking at where the insects are hiding in these sort of desolate type of areas. Staying with urban areas, also the invasive insects. In forest, natural forest, we're looking at the impact of whatever is surrounding these forests, what's happening on the insect inside them. I love working with dung beetles. So looking at, this was at the SKA site when researchers were still allowed to go there. I'm not sure if anybody's allowed to go there anymore, but they sort of abandoned pieces of land over time, and we could actually monitor what the dung beetles are doing in this land abandonment sort of different stages. And it turns out they like it when we're gone. So the other animals, the wild animals, start to come back slowly, and so do the dung beetles. The dung beetles do not like the dung of the sheep. They like the other dung. We can look at what happens when we replace native animals with our domestic animals. So in this case, we saw decreases in diversity, but also decreases in the body size of a lot of these individuals. So the same species, if it's feeding on wild animal dung versus cow dung, it's much smaller when it feeds on the cow dung. It's not doing as well. And there can be various reasons for that. Back in the forest, looking at the canopies, we try to figure out what's going to happen when temperatures start changing in terms of global warming. So looking at a temperature range or a drought range in different forests, looking at what the insects in these tree canopies are doing currently, and see, sort of predicting what's going to happen over time. And as the title there suggests, we have no idea. Every tree species is differently. The insects react differently on each tree species. So when the climate's going to change, we actually, at this stage, can't predict what's going to happen to the forest insects in South Africa. Working on Robben Island, it's always fun. And then also looking with, remember the name, Ophiostomatalis, these fungi that are associated with insects, and a lot of them are invasive. They're associated with insects, and a lot of them are tree killers. Some of the worst insect or tree killers in the world are beetles that associate with these fungi. And some physiological things. So why did I mention all of that and everything before that? Because I have to mention this as well, is I think it life prepared me for what's coming in terms of this beetle. So I don't know if you've seen this beetle before. A lot of you probably heard of this thing before. This is the Polyphagus shottle borer beetle. That little beetle that's in the news that is killing a lot of trees. I think he's a really cute little guy. Looks like a little hedgehog. But this beetle is really, really small. It's only about the size of a sesame seed. And you only need one individual tree in an area that makes a colony, and that will start a whole epidemic and kill thousands of trees. So I call it the perfect invader. So those of you that had class with me would remember that one of your tasks, the fun task we had, is after invasion sort of classes that we had, you had to go out to take the principles of what makes invasive species an invader and build your own invasive species. Take all these characters and you can do whatever and build whatever you want out of that. You just have to justify it with the scientific literature. And nobody could come up with what this guy is. It really is the perfect invader. It's minute, it hides really well, it has a really wide range of food plants that he can live off, seems to have a very wide tolerance for temperature ranges, it breeds fast, it's associated with humans, so just all of those ingredients put together in one little thing makes it easy to invade. So this is a picture, I can't remember exactly where this was, I think this is in Knysna. It's either Knysna or George. So that is a dead oak tree, so the English oaks that line our streets. So that tree, well, that's a couple of years ago. So that tree took about 4 years to die. You'll see one at the back under the White House there, that's dead long before that one as well. So the beetle got in that area around about in 2014, we think, and it's caused a lot of havoc there. So the initial symptoms of this beetle when it infests trees are really hidden. It's a small little hole you'll see in the bark of the tree. That's where the pen is. That's the entrance hole into the tree, and that's on a plain tree. So if you want to see exactly that symptom, just walk across the road here. Victoria Street this year, they started going into the plain trees now for the very first time. So you'll see a lot of it in the next 2 or 3 years. On the right-hand side there, what was that as? I think that's a Harpophyllum, one of our native species. So you see the little holes going in, but you don't really realize what's happening on the inside of this tree. So take off the bark of that tree, you see those gray areas, that's where a fungus is starting to grow and invade the wood of that plant. What's happening on the inside? Well, one of the other interesting things about this beetle, it creates this gallery system. It basically cuts all the water flow in that tree by creating all of these gallery systems in that tree. But one of the other reasons why it invades so easily, and remember I said you only need one individual, that mom in that picture there, that's a foundress female, that's the one that started off the colony. When she starts a colony, when she drills into that wood and builds the colony, if she has not mated before, all of her eggs that she lay would hatch into males. And she'll just mate with one of her own sons, and then the life cycle can continue again. But usually the brothers and sisters, or sons and daughters, will mate with each other before they leave that gallery system. And there seems to be no problems genetic-wise of inbreeding, depression, and those sort of things that you expect in a system like this, which is kind of strange. That's why it's called a fornicating beetle, Uvalacea fornicatus. What also interests this, and why it's sort of right up my alley, is that they don't eat the tree. These beetles are farmers. They use the tree as a field to plant their fungus. So as you can see, in the gallery systems where the beetles are, you'll see that grey lining around the galleries there. That's the fungus. That's what they eat. So the female, when she drills a hole into the plant, she actively plants this fungus in the wood. So she carries the fungus into pockets next to her mandibles, the teeth basically, like a chipmunk. And while she chews, the spores are released into the wood and the spores can start to grow. And this fungus grows into the wood, which is for an insect nutritiously really not good. So they need help to get all the nutrients they need. And that's where the fungus comes in. The fungus breaks down the wood and the sugary substances and concentrates that in the tunnel system for the beetles. And that's the only food that these things eat. So we call it a polyphagous beetle, which means that it has many food, but it actually only has one. It's the fungus that's the polyphagous thing. The fungus eats a lot of different things. There's also a lot of debate on who's farming who, because the beetle and the fungus, neither one of them can live without each other. Who came first? Did the fungus come first and then force the beetle to do its dirty job and spread it? Or was it the other way around? So how did it get to South Africa or around the world? It's invaded many, many different countries, 7 or 8 currently. Or not invaded all of them, but it's been detected there. Even in Australia, as of end of last year. And they have very stringent sort of protocols and things at their borders. Probably through shipping of live plant material, but also the packaging, the dunnages and things that's used for that. And the pallets, really low quality wood. It's not treated, even though they say it's treated. It's definitely not treated. We've isolated some weird and wonderful things from pallets at harbours. And it's not the first time it's gotten to South Africa. We've got molecular evidence also now that we at least have one other or extra import of this beetle into the country. The one haplotype, the one genetic variant is from Vietnam, and the other one is from China. And these two have different fungi associated with them, different species of fungi. So we're only seeing sort of a part of what's to come at this stage. The other fungus, we have no idea what is that fungus would like. So it's a real big problem, and we have to do something about our ports of entry. How does it get around when it's in the country? Well, infested wood is the only way, well, unless one of the beetles flies and sits on your grandma's hat after church and she drives somewhere. But the usual way would be through infested wood. So tree dies, people chip the wood, they take the chips. For some reason we like wooden chips in our gardens as a mulching. If the beetles are in there, they're going to climb out and cause havoc. But you can also go to the nursery if you don't know what to look for, and you can buy a plant with it in already. So this is in a nursery in Sandton. I can't remember the tree species there. But that's a PSHB beetle that's gone into that little sapling. So this wood that's chopped, things like briarwood, is a really effective way of this beetle going around. So this is one example from Lanceria. So that's a pile of wood that came from sand to Lanceria for briarwood at a brewery. And our colleagues in Pretoria sent one little piece of that wood, basically the size of a flora tub of margarine. They put it into a dryer to chase the beetles out. And out of that one piece of wood, there were 44 mature females that emerged there. A lot of males. Inside there, still lots of larvae and eggs that stayed. They didn't climb out. If every single one of those found a colony or had a colony, could make a colony, then within the first generation, about a month later, they could have 1,500 babies already. And it just so happened that this pile of wood was dumped right next to an oak tree, which is one of their preferred food food types. So that's really easy for this beetle to get around in the country. In South Africa, those dots are all the ones that we could confirm. We confirm with molecular techniques because there's a lot of other beetles that look the same. And those red dots, those are the ones close to home. So that's Somerset West, Stellenbosch. So it's spreading quite rapidly in Stellenbosch now. It's been here for about a year and a half, maybe 2 years. Not a lot of people are aware of this, and it's spreading quite rapidly, and the first trees have started to die. The very first dead oak tree has been chopped down in Church Street a month or so ago, in the middle of the night, I must add. And then also the beginning of this year, it's the first time that it's been detected in Cape Town, proper Cape Town, so the CBD area, in Newlands and those areas as well. So massive problems coming our way. So what can this beetle do? So that is a plane tree. Trees we have in Victoria Street here. To 2017, that's the same tree 2 years later. That's just one of the branches higher up. Each one of those holes there is an entire colony of this beetle. So during summer, of each one of those little holes, there's at least 60 or 70 beetles that will climb out of that, every little hole per month. 2 months, sorry. This is 2-3 years ago in George, just driving around taking pictures of oak trees. All of those oak trees are gone now. They should have been removed 4 years, 5 years ago already at the first sign of this. So all the oak trees in George are either dead or gone, dead or dying. Even age doesn't matter for this beetle. So that tree is about 200, I can't remember exactly, over 200 years old. And for the church there, a significant landmark in George. You can see the dieback there. All kinds of signs up everywhere in George because these trees are dangerous. A dead tree is a dangerous tree. You can't just leave it. You have to remove it, which comes at a cost. On that tree, that's just a closer look at the tree. Some people have been trying kind of weird things, putting the tree on drips with all kinds of weird chemicals, which had zero influence. And that's the ultimate outcome of that tree. It's gone. And I gave the same warning to residents in Somerset West 3 years ago, 4 years ago, that the exact same thing is going to happen there, and it's busy happening, and the exact same thing is going to happen here, and it's started happening here. But these are the urban landscapes. We want to know what's happening in the natural forests also. So, I mean, we're really worried about these forests. There's 2 victims or students working in the field there. The middle picture there is of, is it our national tree, a yellowwood? I think so. Those little toothpicks there that you see there, you can't see the holes in a yellowwood. It's quite well hidden, but I stuck a toothpick in some of the holes where the beetles went in. So that's our national tree, and the beetles are starting to go for those also in these forests. And some of the biggest trees we have in those forests are these yellowwood trees. And the picture on the right there, that is even our savannas will not be saved. This is some of one of the common Vachellia species or acacia species that's also being attacked by these beetles. So all in all, in the Tsitsikamma forest in those areas, about 10% of the tree species there seem to be susceptible to these beetles. Now luckily forests are a bit more resilient to invasion, so hopefully we won't see the full force of what this beetle can do, but we're starting to see that on the edges of some of these forests. Some of the Vachellia trees, the kierboom, which protects the forest borders, these are being taken out by this beetle. So we've set up some monitoring plots there. These are long-term monitoring plots, almost like permanent monitoring plots. I think there was about 50 or 60 of these plots that we set out, and we go back every 6 months and reevaluate and see what's happening there to these things. This is some of the preliminary results from, I think it's the first 2 years or 3 years of that data. And we're seeing all the common sense things that we expected to see. So the closer you are to an infested area, the more likely it is that you're going to get infested. Makes a lot of sense. If there's a lot of food around for the beetle, the chances are good that you're going to get infested. Also, the closer that you are to already infested host, the more likely it is. So this, you can start seeing already the connections with those experiments we did with the flowers in the boxes, the disease density, those sort of things. These beetles seek out the largest tree first in the forest, which was kind of interesting. So it's not a random thing. So they're finding the largest trees first, and we suspect it's because they're day flying and they can actually see the trees, where most of the species can't. They're night flying. There's one other species, the red bay ambrosia beetle in the US, that they've done some tests on, and they can actually, there they showed that they actually do seek out silhouettes, the biggest silhouettes. So they aim for the biggest thing in the landscape first. So it's not a chemical signal that attracts them. In terms of these trees in the forest, the further they are away from water, the more likely they are to get the beetle. That might have something to do with stress. And also if that tree has been impacted by something else. If a porcupine has chewed the roots of that thing, or somebody has chopped off a branch to make a path. Those are the type of trees that are first targeted. So they do also select stressed trees. And then less diverse areas. So if you have less diversity, the chances to get this beetle is higher, which is good news for our forests because there's lots of areas with lots of high diversity. It's bad news for our urban environment because we do not have high diversity. So we had to team up with some economists to do the sums for this. My own budget is looking dismal, so we had to get other people to help us with this, working out what the impact of this beetle in South Africa can be. So the unmitigated 10-year impact, so that's if we just do absolutely nothing and let this beetle do whatever it can, it's going to cost the country about 275 billion rand in the next 10 years. That's 0.66 of the GDP of the entire country. And most of that cost, you can see in that graph in the table there, if you look at the baseline scenario, that's in US dollars, millions of US dollars, is in the urban areas, which is going to be us, the taxpayers, that's going to have to fork out that money. That's basically trees that die need to be removed. Who's going to pay for that? You're going to have to pay for that yourself. Forest trees, we're going to lose quite a bit of money there. The forestry industry, the only tree that's in trouble is black wattle, which is also one of the reasons why firewood is a bad idea, because they breed in some of these firewood species. And you can actually spread this beetle by buying infested firewood. And at that stage, the only sort of agricultural crop that we were worried about is avocados, because we knew in Israel and California that they actually go into avocado trees. They cause some damage. It's not an apocalypse, so you're going to lose some money. It's going to cost you something, but the avo industry will still continue. So avos might get a little bit more expensive. That was at that stage. And to sort of preempt in the Western Cape, just to see what we can expect to happen, we teamed up with Hortgrow and looked at some of our important agricultural trees. We saw that these beetles go for things like apples and prunes and apricots and those sort of trees in gardens and can actually kill them in a garden. So we got worried about what can happen in orchards. So we did some screening with the fungus to see if the fungus can grow into these things, and the fungus does well in all of that. So that was our first warning. And literally, it's probably about 6 months after we published that paper on sort of this early warning, the first signs of these beetles appeared in orchards. So what you're seeing there, at the top there is grapevine. So they try their luck. Hopefully they will never make it. So they try their luck, but the fungus doesn't seem to grow well in grapevine. So the beetles don't cause much at this stage, problems there. But things can change over time. There's a new fungus in town. The other picture you see there, that is of a plum tree in an orchard. And each one of those sort of gluey bits is where a beetle went in. And we know that they can breed in these trees. We don't know if they're actually breeding, any one of those are breeding. The chances are that at least one or two of those made it and made a colony in there. And you only need 2 or 3 colonies in a branch to ring bark that branch and kill that branch. So we've got a lot of work and students looking at that particular problem as we speak, actually. So this is a pear orchard very close to us, Somerset West. A pear orchard, that's one of the trees in that pear orchard. So every little dot you see there is where a beetle tried its luck going into that tree. You can see lots of bleeding, lots of sugar. That tree is fighting hard. Just a closer look there. The tree is really not happy. If you open that tree up, You see that. So that's under the bark. You see those dark areas there? That's where the beetle went in. That dark circle around it, that's dead vascular tissue. So the fungus has already killed quite a bit of the vascular tissue of that tree. But you also see those long lines going up and down in the tree, those pink lines. That's the fungus growing. And it grows extremely rapidly in pear trees for some reason, and very, very far up and down this tree, even up down into the root system of these trees. And this is really hot off the press type of things. This is since, I think, last year, Vian. Last year was the first time we actually started seeing this. So it's the first time in the world that these beetles have gone into orchards other than avocados, into these deciduous fruit orchards. So we ground zero. We have to try and find out what's going on here. So into chemical management, everybody always jumps to the chemical side first. With something that's inside a tree, it really is not easy. There's a hole in the tree, so that already stops water and nutrients moving up and down. Then the fungus blocks some of these vascular tissues, which causes even more issues. So you are limited. But we came up with some interesting ways to test this. So we can actually put the fungus in a tree and put the beetle in a tree and then treat the tree and see if that worked. So rather than treating a tree and waiting for 3 or 4 years, before you get the results, 2 months later, we can actually have the results. So on that bottom right picture there, that's a little 3D printed device that we use. We put a beetle in that, and you put it onto the tree, and then you allow that beetle to actually enter when and where on a tree you want it to do that. So there's some of the devices on the left-hand side there on a tree. So that's where the beetles, we let them go into a tree. We tape areas where you put the fungus in that tree. 2 or 3 months later, you open up that branch, and you can actually, on the right-hand side there, you can see we can actually follow the gallery system of the beetles. We can see how far they travel in the wood just by following the fungus stain there. And then you can open up that gallery and actually count on the inside how many eggs, how many larvae, how many whatever. So you can actually follow the development of these things. It would actually be nicer to just do scans of these things rather than do it like this. Do some CT scans maybe. But by doing all of this, and on trees that we treated chemically, we can actually see, did we actually have an effect on what's going on inside in the trees there. So these are some of the chemicals that we tested. So this is just a number of new attacks on the trees. So this is 2 months after various treatments. And 2 of those treatments, sort of trunk spray treatments, reduced the number of attacks significantly. However, I have to caution, significantly in this case doesn't mean much, because they still get in. You don't want any beetles to get in. So we've reduced it by about half the number of attempts they make onto a tree. And we've used very high concentrations. In fact, illegal levels of concentrations that you will not be able to apply anywhere. It's really the highest we can go with these things without being phytotoxic. If you look at those galleries, when you put the beetles in, it's looking at how many of them actually made colonies versus the ones that didn't make it. If you look at the controls there, so the success is the blue, so the number of colonies that were successful. The controls, we got about 70-odd% successful colonies just by doing nothing to the trees, and then with all of these treatments we could reduce the number of colonies, the success. But again, it's about by 30 to 40% if you're lucky, using high concentrations of these chemicals. It's not enough to stop this. You're just delaying the process by doing this, and it's going to cost you a lot of money. Fungal growth, the only one that worked on the fungal growth there was the propiconazole. It's the only fungicide that we also tested there, so it's no surprise there. So the take-home message from the chemical treatment is the same thing that we had to do this. It's been done in California. So the same thing that they found is it's not going to work. You're going to waste a lot of money. You can slow things down a little bit, but that's about it. So as you can understand, there's a whole lot of projects and students working on this particular problem. This is just some of the projects at Stellenbosch University that I'm involved in. I'm not supervising all of these projects. I'm not the main scientist involved in all of these things. But these are some of the studies at Stellenbosch University, looking at what's going to happen to the fruit trees, looking at some biological control options. It works in the lab. As soon as you try to do it in the field, everything falls apart. It's a difficult system. More chemical control options, looking at epidemiology, looking at whether you can use satellite data to track the movement of these insects through the landscape. So all kinds of things. Yeah, so with that, those are the three stories that I would like to tell of the interactions between different kingdoms of life and what they can do and how they influence us. This is a word cloud of all my co-authors. And you can see there's a couple of names that jump out there, sort of people that I've walked a long, long journey with and that helped me quite a lot. You'll see Mike Wingfield, Leon Dreyer, Pedro Kraus, Brenda Wingfield, Janneke has done quite a lot of the microbial work and genomics and things like that. Kenneth Weberlander, he's probably online. We've sat along next to a lot of fires contemplating questions that we never got answers for, and a whole bunch of other people. For those of you that's authored with me, your name is there, but it's very small. You need to phone me. We need to do a little bit more work together. And just like with these other organisms, none of this work can be done in isolation. You have to build networks and large networks with this. And these networks are not just the science community or colleagues, but also family and friends. And all the students, I did not list everybody here, past and present. Thank you very much for your hard work. Thank you for dealing with me and my idiotic ideas sometimes. I go out and do this just for interest's sake, whether you understand it or not. And for me, wasting a lot of your time. At least you enjoyed it, I hope. My parents, thank you very much for letting me do what I do, even from letting me sit on an ant's nest when I was a baby, just so that I can learn not to, maybe, I don't know. But thank you for giving me the space to do whatever I wanted to do. My brother, the back there, thank you. He had to endure thousands of hours turning over rocks and walking and looking for bugs, which he was not that interested in. In fact, he kills them for a living. At this stage. My kids, you are here because I want to tell you, don't do what I do if you want to make money. It is fun, but you're not going to get rich. My grandparents, they're not with me, but I would love to say to them is, yes, this is actually a real job. And then my in-laws and non-sciencey friends, I hope you have a little bit more of an idea of what I've been keeping myself busy with. And I'm probably not going to explain any of this again. So if you didn't listen, that's too bad. And then Gabby, it is really rare to find somebody that you can walk a journey with, that we can talk to for hours, sometimes way too late at night, about science, about life, about everything in general. And thanks for making it fun, the journey. I think that is what I want to say. Some of the funness, I'm pretty sure I forgot a couple of logos. I ran out of time. Thank you to everybody that's paying me to play with all these bugs. I'll leave you with 3 of my favorite newly discovered dung beetle species. I just love these creatures. Thank you very much.
Ladies and gentlemen, I'm delighted to say that my tribe made a minor appearance in the lecture this evening. I'm from The Economists. I speak on their behalf, and I will say something about that in a minute. But we're here to celebrate something else. So an inaugural lecture is a milestone in an academic's career, and it's an opportunity for a full professor to speak to his peers and also to the rest of the public, and to share something of this lifelong passion which brought them to the highest academic rank we have in the university. And we call it an inaugural lecture because we use the Latin word that reaches back to the idea of predicting what is to come. That's the root of the word. And you will also have seen that this evening. So it's not just a a tale of what the professor did, but it is a prediction of where the professor and the professor's team is heading, because modern professors work in teams, as apparently do beetles and fungi and mites. So, but I also want to say something else. I felt this evening that we were in the hands of a very crafty storyteller. And he pulled us first in with a happy story, right? So first we had these protea mites, right? And they're cunning. They're inside a protea, and we don't know how they get there, and then we discover, and then we also don't understand how they can travel such vast distances. And it turns out they've got bigger mites, and those have birds. And it's a beautiful story of intended and unintended collaboration, which is a story that I really warm to, because the tribe that I'm from, we're all about collaboration. We think that in the tale of Robinson Crusoe, there's nothing interesting that happens until Friday arrives. It's only then that stuff gets interesting. And it's like, when the bigger mites arrive that we get really interested in the story. So that's the first happy story. Then things get a little bit darker already when we get to the sexually transmitted diseases. And the astonishing discovery that some bees have preferences for diseased or non-diseased flowers. And it made me think that one of the great founding figures in all of social science, an unheralded figure because of the scandal that he caused, is a guy called Bernard Mandeville, a Dutch writer of the early 18th century. He wrote a very famous, scandalous tract called The Fable of the Bees, which was all about how bees, well, in the allegory that he wrote, we manipulate their preferences to be more virtuous in the early 18th century understanding of it. That causes everything to go very badly wrong. We have to leave them to their natural choices. So your description of the natural preferences of these bees reminded me of Mandeville. But then things got really dark in the third episode this evening. So then we got to the shot-hole borer beetle, described as a fornicating beetle, a perfect invader with incestuous habits, which apparently already lives in Victoria Street. I'm alarmed by this fact, because my day job is to look after the campus, which apparently is being eaten as we stand here. And I wish you had, less glowing CV for this perfect invader. And so we've heard of ways of trying to combat it. We hear that it's going to cost us 275 billion rand over the next 10 years. And for the audience, just to give you a frame of reference for that, what that number means, that is 60% as much as our country will spend on universities over the next 10 years. So it is a truly large amount of money that we're going to divert, apparently to just cut down the trees. It's not to stop the beetle, that's just to cut down the damage of the trees. And the chemistry won't help. But I am encouraged by the fact that Professor Roets and his team is now working on other solutions. I can assure you that you got my attention this evening. And I also say that to the dean, because we have to find practical solutions to this problem. And I'm going to close off by saying that it is really encouraging for us as a university community to see a top scholar working not just on what he's passionate about, but also on topics that matter for us as a society. Francois, it's a great honor to have you as a part of our professorial community in the university. Congratulations with your inaugural lecture, and good luck with the research.
Thank you so much.
Thank you, Professor Du Plessis. Please, guests, can you please stand as the procession will leave now, sorry. Italy.
Right, which way do we go? That way.
This way.
Okay.
Yeah. Thank you for attending. Enjoy the refreshments.